So today we're talking about cell organelle structure and function. We're mainly going to be paying attention to the structure of cell organelles today. So to start us off, we want to think about what is a cell. And to kind of guide our thinking, I want you to keep in mind these two ideas, unity and diversity. When we're thinking about cells and cell types, we want to think about what do they all have that's the same, what's similar, and what's different as well. Okay? So, to the basics of it, what is a cell? Well, a cell is the structural and functional unit of all living organisms. So a structural and functional unit of all organisms. We really want to think about, first of all, what are all cells made up of? We've talked about all of our biochem, we've talked about all these different molecules that are used to make up different portions of cells, okay? And that are important to all living things. And now we want to start putting those pieces together. So the first thing that we talked about when we were looking at a cell was the outside of the cell. So all cells are going to have a partition or a barrier known as the cell membrane. Okay, we should be very familiar with the cell membrane now. Remember that it's a barrier between the outside water and the inside cytoplasm of the cell. So all cells are going to have a cell membrane. Because of the chemistry of the cell membrane, of those phospholipids, wanting to be hydrophilic or hydrophobic, or actually being hydrophilic or hydrophobic, having the cytoplasm inside of the cell was important to that. So remember that those phospholipid heads were hydrophilic. They like touching the water. Well, inside the cell you have this watery jelly stuff, the cytoplasm, okay? It's a jelly-like substance or jelly-like fluid inside of the cell that's mostly water. And a lot of times this is going to provide some support for the cell as well. It's going to hold up that cell membrane, okay? And so we have those hydrophilic heads of the phospholipids that are going to touch that cytoplasm. Now within the cytoplasm we'll have lots of different structures, lots of organelles, tiny organs inside of the cell. But the main thing that all cells have to have is some DNA. Remember that DNA is made up of nucleotides, it's a double helix, and it's the code, or contains the code, for proteins, for building proteins, for assembling those amino acids. So all cells will have that. And because all cells have DNA, which is the code to make proteins, all cells also need to be able to make or build the proteins. So we have a structure within the cell called a ribosome that allows us to build the protein. So all cells will have this, from bacteria to fungus to protists. They'll all have these four main structures. One other thing we want to keep in mind is what makes one cell different from another. This is the unity of it. These are all things that cells have that's in common, or that's the same. And so the differences between the cells, if you're looking at this picture, we have different cell types that are affecting different parts of your body. Okay? These are actually cells that didn't form properly for some reason. That's causing heart failure or arthritis or Parkinson's or diabetes. So something went on with these cells. But if you look at the very first cell at the top, notice how it can divide into these many cell types. So a cell like this is known as a stem cell. And you just want to know that a stem cell is undifferentiated. that it can still become any of those cell types. Right now, all the cell has is a cell membrane, cytoplasm, DNA, and ribosomes. But the DNA and genes within that DNA aren't turned on and off yet, okay? For one cell to be different from another, it needs to make different proteins. It needs to have more specialized parts or functioning units. So a stem cell is not yet differentiated, not yet specialized, and still has opportunity to become any of those cell types, okay? And it still has all four of these structures, but basically that's it. Moving from there, we're going to continue looking at what do all cells have in common. Now we're actually going to compare some cells. The main thing that's going to allow us to compare the two main cell types is the fact that they all have DNA. And we can actually say that DNA is universal to describe that fact, okay? So all cells contain DNA, whether you're bacteria, a plant, or an animal, or a protist. And the main thing that's going to separate the two types of cells is where is the DNA held. So our major division is going to be based on where is the DNA. Okay. So when you're looking at this image, we have two major cell types. We have prokaryotic cells and we have eukaryotic cells. The word kary, K-A-R-Y, in both of them is related to the nucleus. Okay, it means nucleus. But prokaryotic is going to mean before the nucleus. Okay? And eukaryotic, eukaryotic, means true nucleus. So basically the major division depends on whether or not you have a nucleus, an enclosed membrane-bound structure to cover and protect your DNA, or if you just have a region to hold it in. When we're looking at prokaryotic cells, those are going to be mainly bacteria. A few things we need to keep in mind about bacteria is that they are smaller, okay? And they're actually a lot more simple than our plant or animal cells. They have less going on. They have our four basic things and maybe a few more exterior structures. But when you're looking at your prokaryotic cells, a few other things to keep in mind is that it does have DNA. However, the DNA, usually they just have one circular chromosome. And remember, a chromosome is just how the DNA is packaged. Okay? So this chromosome is coiled up, coiled, and folded as well. And actually, it's going to be contained in a region known as the nucleoid. So instead of having a separate compartment for the DNA, it's all condensed in one area of that cell. So main thing here, we have no membrane surrounding the DNA. Okay. Some positives to this situation is that it's the accessibility. Okay. Because our DNA isn't held within a separate compartment, we can get to it really easy if we need to. But the downside of this is because there's no separation, there's going to be no protection. And it's also going to be less organized. Okay? It's just kind of clumped up into that region. On the other hand, with our eukaryotic cells are going to usually have DNA that is more linear. And they actually have more DNA. Okay? Or a larger amount of DNA. Not that the DNA is bigger, but they have a larger amount of them. And inside of these eukaryotic cells, we're going to have more complex structures. Such as the nucleus, is a membrane bound structure, which is around the DNA, or that covers or contains the DNA. Okay. And so the last thing we actually want to look at are some specifics about our bacteria cell. So when we're looking at the bacteria cell, we want to look at some specific structures that it contains. So what structures do prokaryotic cells have, and how do these help bacteria function? For prokaryotic cells, those are going to be our bacteria. And we actually have two types of bacteria. We have our EU bacteria, or our true bacteria. These are generally the bacteria that will make you sick. And we also have our archaebacteria. And the archaebacteria are actually known as extremophiles. They can actually survive extremophiles in very extreme conditions, places that are really hot, or really salty, or maybe have a really extreme pH. Okay? So looking at these structures here, the first one that we want to talk about is the capsule. Okay? The capsule is made up of peptidoglycan. Pepti referring to protein, glycan referring to the sugar. Okay? And this is going to be a support structure for the cell. Oh, actually, I'm so sorry. That's actually for the cell wall. Okay, so number one will be our cell wall. Number two, we'll actually put as our capsule. Okay, and our capsule actually is the most exterior structure and allows it, as in the bacteria, to stick to its surroundings. So that might allow it to stick to your cells to make you sick. Okay? Now, of course, the bacteria is also going to have a cell membrane, which is what contains all of its structures. Okay, we already know about that. The nuclear material will be our circular chromosome. One thing there with nuclear material, we also have, if you'll draw a little circle and write in plasmid, we'll do that as number 3. Number 3, the plasmid is just extra chromosomal material that later on we'll learn that the bacteria can exchange. Okay? It can actually be traded. Can be traded. And this actually increases the variety of the cell or of the DNA that the bacteria can have. Other structures here, the flagellum allows for more movement. Not all bacteria have it, but a lot do. Same thing with the cilia that you see on the outside of the cell. Also allows for the movement of the cell. Inside here, we also have ribosomes that allow us to make proteins. Nucleoid, which is the region. Cytoplasm, which is the liquid inside of the cell. Okay? So that's our basic covering of our prokaryotic